We're live.
Yay!
Yay. There you go.
Woo!
The surprise feature from George, as you can see.
That's
Very
old Tabby
nice.
Cat.
All right, let's get started, boys.
All right. So what are we reading today?
Cell signaling.
Cell
We're
signaling?
going over cell signaling. A great 480 topic.
Sure is.
Oh, yeah.
What's the name of the article?
The name of the article is called Multiple Signaling Pathways Involved in the Metabolic Effects of Insulin.
Oh.
Okay. So this is from the University of Bristol, School of Medical Sciences in Bristol, the UK.
Oh, okay. From the American Journal of Cardiology. Are you a robot? No. Okay, I'm sorry, go ahead.
All right. You guys want the abstract? You want the quick rundown? Or?
Yeah, do the abstract. We can get through the gist of it.
All right. Colton, how about you read it?
Okay. The abstract. The metabolic effects of insulin are initiated by the binding of insulin to the extracellular domain of the insulin receptor within the plasma membrane of muscle and adipose and liver cells. The subsequent activation of the intracellular tyrosine protein kinase activity of the receptor leads to autophosphorylation of the receptor as well as phosphorylation of a number of intracellular proteins. This gives rise to the activation of RAS and phosphatidylinositol 3 kinase, and hence to the activation of a number of serine threonine protein kinases. Many of these kinases appear to be arranged in cascades, including a cascade that results in the activation of mitogen-activated protein kinase, and another that may result in the activation of protein kinase B, leading to the inhibition of glycogen synthase kinase 3 and the activation of the 70 kilodalton ribosomal S6 protein kinase, P70 S6 kinase. We've explored the role of these early events in the stimulation of glycogen, fatty acid, and protein synthesis by insulin in rat epididymal fat cells. Comparisons have been made between the metabolic effects of insulin and those of epidermal growth factor. Since these two agents have contrasting effects on P70 S6 kinase and mitogen-activated protein kinase, the effects of wortmannin, which inhibits phosphatidylinositol 3 kinase, and rapamycin, which blocks the activation of P70 S6 kinase, have also been studied. These and other studies indicate that the mitogen-activated protein kinase cascade is probably not important in the acute metabolic effects of insulin, but may have a role in the regulation of gene transcription and hence the more long-term effects of insulin. The short-term metabolic effects of insulin appear to involve at least 3 distinct signaling pathways. 1, those leading to increase in glucose transport and the activation of glycogen synthase, acetyl-CoA carboxylase, eukaryotic initiation factor 2B, and phosphodiesterase, which may involve phosphatidylinositol 3 kinase and protein kinase B. And 2, those leading to some of the effects of insulin on protein synthesis, formation of eukaryotic initiation factor 4F complex, S6 phosphorylation, and activation of eukaryotic elongation factor 2, which may involve phosphatidylinositol 3 kinase and P70 S6 kinase. And finally, 3, the leading to the activation of pyruvate dehydrogenase, which is unique in apparently not requiring activation of phosphatidylinositol 3 kinase.
Fun. That is a lot.
I was going to say, did you guys even, yeah.
That's a lot of information right there.
I know
Yes,
what most
it
of
is.
that was, but now I have to re-skim it myself to put it together.
Okay, cool. Well, let's talk about it. What about what do you guys remember about glycogen synthase?
Glycogen synthase?
Yeah. It makes glycogen,
Yeah.
right?
Yeah.
It's what you need to start making glycogen.
Right. So, in the world, we put glucose in ourselves. We drink it in our energy drinks. We eat it in our cake. We eat it in our cookies.
You get it from almost everything.
We get it from literally everything. We get it from fruit. Great place for natural fructose.
Yeah, sure.
And glucose.
Sure.
So, what the body does is when there's an excess of glucose, it decides to transform those into a network of interconnected glucose molecules via an alpha 1,6 glycosidic linkage.
Yep.
Good. Those form in little strains and string off from one each other in a branch network. That glycogen can then be broken down by the body during periods of starvation so that it can go through the whole process of glycolysis, breaking it down, producing your 30-ish ATP, and that gives you energy. So what insulin does, it activates pyruvate dehydrogenase. You guys remember what pyruvate looks
Yeah, I
like?
remember that.
Yeah.
Pyruvate dehydrogenase.
Who doesn't remember what pyruvate
That's
looks
a big
like?
one.
But pyruvate dehydrogenase basically is removing a water from something. Something that's dehydrating is removing the water.
Dehydrogenase is remove water.
Exactly.
Yeah, there you go. Yeah.
Just so we're all clear.
It's an enzyme.
That's
So
what they do.
they remove a little H2O molecule from something that usually had a couple of those things integrated, mostly through an exothermic reaction.
For which one?
Dehydrogenase. It releases energy. It's a
Huh?
favorable interaction, just like breaking
Yeah.
down a polypeptide.
I mean, yeah.
Peptide breakdown is a usually exothermic process. It releases energy. It's spontaneous. There's not a huge initial hump to get over.
Yeah.
So, what else can we take away from this? Subsequent activation of the intracellular tyrosine protein kinase. Everybody know what tyrosine is? I don't think
No, I think
anyone watching knows what tyrosine is yet, but tyrosine's pretty cool. Colton, explain what tyrosine is. I'm putting you on the spot. I'm sorry.
It's an amino acid.
Yeah.
It's aromatic and has a phenol group on it, and it can be phosphorylated by removing the hydrogen on the alcohol.
Oh, you can?
Which is on the phenyl ring.
You gotta push that one. Sorry.
You're messing around with
Oh,
the
yours
chairs.
is unplugged.
It's
Mine's
amino acid.
unplugged.
It's
Oh.
used a lot in protein.
No, it is. It's the amino acid that's used very much.
Yeah.
But mine has light.
That's the other one.
Wait, no,
Colton's
this isn't the
works.
other one.
I think
Colton's
this one's
works.
just
Mine's broken. No, but mine can't be broken. Look, it's got the thing.
Don't know.
I'm in a chair that's broken, kind of. So his goes back. Mine's cup holder lights up right here.
You gotta stay focused. Anyway,
Heck
that's not
of
the
it.
point though. Anyway.
All right, whatever.
No, yes, that's what tyrosine is.
Tyrosine's pretty cool, yeah. So phenyl is a benzene ring. Attached to it is an OH group. Very useful amino acid, especially in catalytic pathways.
In DNA, there's a lot of tyrosine.
Oh,
we're
yeah.
talking about. For
Lots
the-
of coordination, lots
Yeah.
of grabbing things. It's
Mhm.
pretty nice. Autophosphorylation. So for something to be phosphorylated, usually it needs an oxygen somewhere. It needs to be either an alcohol, needs to be attached by a glycosidic linkage or something, but phosphorylation involves the attack of a phosphoryl group by that oxygen as a nucleophile, pulls off the phosphorus with 3 other oxygens, and then it becomes phosphorylated. That's when it could be considered phosphorylated. I draw a picture, but it's a little complicated.
Oh, I forgot my whiteboard. Just gonna bring that.
You should bring the whiteboard next time.
I'll bring it for the 3rd episode.
We
Draw a little
got that, but
Yeah.
anyway.
attack.
Phosphorylation is used in biological systems to activate or deactivate.
Because the energy.
proteins.
Phosphoryl groups are highly energetic, right?
Oh, yeah.
Negative 30 kilojoules per mole.
That's
Mhm.
true.
So you get a lot of energy when you're using them for certain things, which is why I think they're so prevalent.
That's
Right.
true.
I think so. Oh,
Yeah.
Who's viewing us right now? There's one viewer.
I don't know. Some people are
Maybe
viewing
it's a bot.
I think it's me.
No, it's not a bot.
Oh, duck on it.
That's okay. So what were you looking at after that?
The paper?
Yeah.
So
You went to the abstract.
Yeah, we're on the abstract right now.
Oh, I was going to ask you a question about that. What was my question? It was at the end, right here, actually.
at the end.
Well, yeah, it said that the activation of pyruvate dehydrogenase is unique and doesn't require the activation of phosphatidyl inositol 3 kinase. Sorry. So
Inositol?
it doesn't require its activation,
Okay.
which is interesting.
I don't
So
know
it's
what inositol is.
phosphatidyl inositol. You can look it up. It's fine.
I don't know what that is at all. I don't remember hearing that anywhere.
Might not have gone over it yet.
Okay.
Title inositol. I'll read you the textbook definition.
Well, phosphatidylinositol is a lipid. It's like a
Yeah, phospholipid found in cell
That's what it is,
membranes that
yeah.
acts as a vital precursor for intracellular signaling molecules.
so it can be phosphorylated, and that is used for cell signaling pathways
But that's not required.
for protein or pyruvate dehydrogenase.
Yeah, it's interesting.
This is interesting. Yeah.
Actually, it's kind of interesting. Can be obtained through certain foods. Oh, okay. It's taken as a supplement, huh? Interesting stuff.
This is interesting stuff.
What else about the article were we interested in talking about? Did you explain what insulin was?
Maybe.
I don't remember. I don't
remember what insulin is. But
Insulin is.
I do remember what insulin
It's
is.
a primary messenger.
Yep.
So it's
Insulin, norepinephrine, adrenaline.
yep. They're steroidal. I'm pretty sure, right?
Yeah.
And that means their structure has 4 rings in it, and it's based off of cholesterol.
And they're competing because one's driving glycolysis, one is driving gluconeogenesis.
Yeah,
Mhm.
yeah. Adrenaline
Yep.
and insulin, they're competing with each other. Yeah. They use similar. They can sometimes use like alpha receptors and then beta receptors.
Yeah. When thinking about it, you always think insulin, glycolysis, norepinephrine or adrenaline, gluconeogenesis.
Yes.
Though I don't like adrenaline, I like norepinephrine because it sounds more like my name.
Norepinephrine is definitely what he wanted us to remember.
Well, it sounds more like my
Adrenaline.
name, so it's pretty easy to remember.
Oh, really?
Yeah.
Okay, cool.
That's how I always remembered it. I was like, oh, that's right. That's right.
Yeah.
Yeah.
Nice job.
Fun
This
stuff.
cups. This one.
We're talking about insulin now. Interesting.
Yes.
Good old insulin.
Insulin.
Insulin?
Insulin.
That's not insulin. That's quantum physics. It says that everything.
Oh, we should do quantum physics, dude. We should really talk about quantum physics. You don't want to talk about quantum physics.
Why don't
No,
we talk about
I
quantum
don't
physics
like quantum
on a
physics. It's annoying.
biochemistry podcast?
Yeah, that
But
too.
there is important stuff in quantum that we need to account for in biology.
Well, listen, I read a title just now that says quantum physics says that everything observed is affected by observing, watching it. What?
Well, yeah, that's back to the old. That's the paradox where you can't.
It's like
see things.
Schrodinger's cat experiment.
I feel like people just make stuff up when they do quantum physics. I don't know.
Well, they
I mean,
have to.
yes, yeah.
There's no other way to do it.
Yeah, I'll stick to biochemistry then.
You ever
At
pull
least
up
we can
those
see
stock
it.
images of quantum mechanics and you try and go through it and realize they don't even know what they're doing?
Yes.
They're pulling from source material
Exactly, that's why I'm
across
like,
the
what is
webs. And it's
going
like,
on
what
here?
are you doing?
What are you doing? I don't know.
They're just pulling a bunch of size. They're pulling a bunch of partial second derivatives. And
I would understand it. I took PChem, but it's just like I just don't see the purpose.
well, they're pulling out matrices, row echelon matrix, like higher order matrices right here.
Yeah.
It's
That
crazy.
stuff I haven't done yet.
I have not done
I gotta do higher.
anything
math.
remotely close.
Anyway, back to our biochemistry article. I might grab some Dr. Pepper.
We should probably talk about the insulin pathway a little bit.
Let's talk about
We
insulin
should. Let's talk
pathway.
about the insulin pathway. Go ahead, Colton. What do you want to talk about in the insulin pathway?
Well, I guess we might as well start at the beginning. Right? It's
Start at the beginning.
so whenever you eat food
You want this, Michael?
and
Oh, no, the rest of my Sprite.
there's an elevated
You want some of this?
presence of glucose
Yes.
in the bloodstream,
It's the bloodstream, yeah.
your body releases insulin as a way to tell the cells to bring in glucose into so they can use it for glycolysis to produce ATP. And so insulin will bind to the receptor tyrosine kinase, which has a receptor on the outside and then the tyrosine kinase on the inside, which once the insulin binds, it has a conformational change, and that causes the tyrosine to be phosphorylated, which then gives a binding site for
Mr.
Other
Mason
proteins?
wants us to talk about electron tunneling.
electrons.
This is something that we went over in OChem. He knows.
Talk about electron.
This is a if you know, you know kind of thing. Because I know one of us went over this.
At Mason Cundy 5985. I'm just curious why we would talk about electron tunneling during a biochemistry podcast, but we can do it for you.
Yes.
You can do it.
Electrons are weird. That's pretty much all.
Electrons
All we
are
need to know.
pretty weird, yeah.
I remember going over this very briefly, and I know he knows, but let's pull up a very simple diagram. This is like some wrinkle in time stuff here.
Yeah, it's electrons. They do pretty much just
They-
wrinkle
yeah,
down.
they
Kind
wrinkle.
of just are here and then they're over there.
Yep. It's like a cat. They just do whatever. You don't have to worry about them.
That's true. That's
They
true.
behave a lot like just fairies or leprechauns or.
He said it
magical
was
creatures.
just a quantum mechanics meme. Oh, okay.
Oh, thanks for the gifted sub.
Was that
Whoa.
sub?
I don't know what
number.
that
I think
is.
he's the number one
He's the
poster.
number one poster. I don't know what the. I'm getting mixed up with Twitch, YouTube, and
No, gifted
everyone
subs
else.
pop up on
It
the
would pop
screen.
up.
Mm.
Yeah. Which is how Twitch
He's not
works.
going to give us that. I don't even know how that works.
I mean, we'll take the money.
I mean, yeah, we'll take the money, of course. Hey,
None of us really know how the internet works. He
I don't
does.
think
I
you
know
know.
how the internet
I
works.
don't know how the
I
internet
think
works.
Colton also probably knows how the internet works. I
I
think
do,
it's just
yes.
you.
I think I'm just the
Yeah.
Luddite
That's
among
okay.
you.
That's okay. But no, what were you saying about the insulin pathway, Colton? The tyrosine is phosphorylated?
It phosphorylates itself. It's
Yeah,
autophosphorylation.
it's autophosphorylation.
Yeah.
There you go.
And
About that.
yeah, and then other proteins recognize that it's phosphorylated, and then they pop on, and that causes a signaling cascade, as I said in the paper, that activates different systems for phosphorylation, like protein kinase B.
Yep.
The phosphatidylinositol 3 kinase, is that stuff, and that can activate more proteins to, process glycolysis and glucose.
Sounds like you really know your stuff, Colton.
I do.
That's good.
He always did very well in these kinds of classes.
Yeah, he's very biochemistry. oriented.
He's a chemist. Yes,
Mhm.
you were good at quantum too.
Yeah.
At quantum?
He enjoyed all of it. Yeah, well, yeah, the
Oh,
Bechem?
Bechem too, and one.
PChem wasn't bad.
It was not bad.
I think it was worse in the moment, but that's only because we had 2 exams a day.
That was pretty awful.
That was
Oh.
the part that made it difficult.
That was
Yeah.
very rough to handle
Yeah.
because we'd have an exam at 10 a.m. and then we get 5 minutes to just mentally prepare for the biochem exam, which of course was always worse. So we couldn't get the worst over with and then have an easier one. We had the easier one first and then the harder
Yeah.
one later.
That was a fun time.
It was always the worst.
Outside of that, though, the class itself I don't think was too difficult.
No, I feel
No.
like if we just didn't have exams on the same day, we could have done better in both.
Yeah.
But it's just the way it works. And even our profs, they said the same thing. Well, that's life. Got to figure it out.
Good old Yao.
Man, I love Dr. Yao.
Yeah, he's a good
Good.
guy.
He's great. Awesome guy.
Great guy.
If anybody goes to Eastern, pick out classes from Dr. Yao Hanzanubo. Great stuff.
Don't say that because
You're
they're
gonna-
going to ignore all the rest of the teachers.
Yeah, well, no. There's plenty of
They
ways
all teach
to.
the same stuff. Yao's not bad.
Yeah, you're gonna learn everything regardless. But if you want someone to, work under, do TAing for their labs or something,
Just
Okay,
just pick any teacher from Eastern and go there. You'll love science. That's what you meant to say.
that'll be fine. Yeah.
Yeah.
I've never had an issue with any of the chem profs up there. None.
No, honestly, no.
They're all
So-
good.
They're all pretty
You're
good.
you're good with everyone. We're not gonna pick names here on this
Yeah.
podcast.
Unless it's Dr. Ben. You should definitely take all his classes.
There he goes.
Yeah. He's just the best of the best of the best ever.
He's very good.
Very good. Yeah. Very funny guy.
Very funny. Yeah.
Very funny.
Anyway, back to electron tunneling.
Yeah. I don't think he actually wanted to talk about electron tunneling. He said it was a meme.
Go back to self-signaling.
Yeah, back to cell signaling.
Come on. I want to learn more about quantum tunneling.
We
I
could-
know, but we should probably
Third times other than
Maybe we should
right
look
now.
at chemistry memes.
we should stick to cell signaling. Cell signaling.
I
is
thought
great.
Norm-
Sorry,
gonna
I'm
be
trying
the
to make
most
I'm trying to make the
distracted.
podcast funny in some way.
He thought I was the one who was going to be distracted. See,
Yeah.
No, I'm very distracted. I
he's
get
seeing
very
my
distracted.
professional side.
Mm.
It's not even very professional at all. But he's trying. Like what? I don't think professionally, guys. Sorry, I have to laugh every 30 seconds, and then I go immediately back to professional stuff, then go back to laughing. It's like
It's a vicious cycle, and
Yeah.
he's great at it. So
it's very fun.
That's
Mhm.
true. Yeah.
you should
Oh,
definitely
cool.
follow this guy on LinkedIn or YouTube or one of them.
Yeah,
If she posts on LinkedIn,
I never made a LinkedIn profile in my life. I did it just to talk to this guy. So
wow,
he's got a great page. So
I do.
just,
Nice.
just look into his LinkedIn.
I spent like 15 minutes on it. It's great.
You know, I spent about as much time online. Too.
Yep.
About
Yeah.
20 minutes of just typing random garbage.
Yeah. Well,
And then I got some guy, some PhD in the comments going like, dude, your LinkedIn profile is made for academia. You're supposed to make it for jobs. You have a terrible, terrible introduction.
really?
It was weirdly focused to me. I know he didn't actually respond to me that way, but it was in my
No.
feed, and I'm thinking
Oh, that makes sense.
maybe this he's trying to contact me. The thing is, I don't know how LinkedIn works. I don't know if he actually is talking to me or not. I'm just gonna think that he is.
Okay.
So,
No, that
yeah.
makes sense.
If you guys see comments under my LinkedIn profile, you'll know who it is.
I think ResearchGate's more for academia than LinkedIn. Anyway, we're way off topic now. Go ahead, Colton. Continue your self-signaling talk.
Cell signal.
You were on the insulin path. You didn't finish it. Did you?
Yeah, I mean,
Oh, pretty much.
I got the gist.
Okay, they got the gist. You guys got the gist.
You got the gist.
Yep. Okay, that works for me. So now that we've gone through self-signaling, let's go back to this article. Oh, this isn't the right chat. It's an article. Here it is. Multiple cell signaling effects. Signaling pathways involved in metabolic effects of insulin.
Yeah.
Well, we kind of knew that, though. What kind of question is that? Who chose this article?
Wait, we can get full access.
Oh, we can. We have to sign in to research. No.
Oh, you do. Okay.
Yeah, it's one of those sign in things.
So anybody's going out to look for these papers, you're gonna have to go through a bunch of hoops to even just get access or even get a PDF.
It's a process.
Usually, you can see the free PDF on the side near the right-hand side, and yeah, it's rough to deal with. I wish it was easier, but they don't make it easier.
So the first event after the insulin is the binding of the extracellular alpha subunits to its receptor, and then the activation of tyrosine protein kinase activity. That's what we were just talking about.
Exactly.
Tyrosine's really big. That's interesting. I've always liked histidine better.
No, my favorite is lysine. I just always like the lysine. It can be a nucleophile and electrophile, really.
That's true.
It does a lot of coordination chemistry, but I was amazed at just how well lysine does certain chemical things back on the first, like the third homework that we've had in 482. Used the catalytic lysine residue to phosphorylate or to alkylate was it a thymine or was it an adenine? It was an adenine. No, it was a guanine. It was an alkylation of guanine. Used.
Oh yeah, you're talking about the homework for
lysine.
this class.
Yeah, exactly. Use some kind of catalytic lysine residue, I think.
Alkylation.
Yeah, pretty cool stuff.
What's your
Signaling
favorite amino
pathways.
acid, Colton?
That's tricky. I mean, cysteine is pretty fun. You can do cross-linking with sulfur.
Sulfur disulfide bonds.
You know,
What about serine? Everybody kind of leaves serine behind. He's just not cool
it's
enough.
not that special.
It's not that special.
Exactly. Yeah. It's just
Serin's
an
pretty
alcohol.
standard.
Yeah.
Arginine's pretty cool too.
Proline's cool looking because it's a loop. Doesn't really do much.
Yeah, but it's
But-
just a loop.
Yeah,
Proline,
it's cool.
I guess.
How did proline form its peptide bond again? Was there a difference there? Was there a difference in how the peptide bond was created with the proline? Okay,
I don't think so.
probably right. You know, I don't even know about that. I'm gonna check that because I want to see polypeptide chain of prolines.
I mean, it's just a secondary amine, essentially,
Yes.
making an amide. So wouldn't be
Here,
that
Colton,
different.
while he looks that up, why don't you look through this and pick out a question we can talk about from this? That's the exam 3.
Okay.
Oh,
Or
geez.
Maybe 4.
That's my bad exam. That's my.
Well, we don't have to read your answers.
Oh, well, he did. He crossed that off, but I was very proud of the next page.
You're proud of this one?
No, not this page. The next page.
He's silently judging.
I
him. You're proud of this
literally,
one?
literally got every single one right.
Oh, yeah.
That was pretty good. Yeah, it was just a numbering thing, but you just had to remember all the pathways of what was it? Phospho phytosine 3 kinase, PI3K. This was insulin signaling. So.
Phosphoinositol 3.
Oh my gosh!
You didn't know what that was?
I didn't know. No,
It's
dude, it's been a year, more than a year.
Jesus.
At this point, it's a year and a week.
I knew I heard about it before.
I mean, I probably just abbreviated it in my brain PI or something, but
Probably.
the inositol, it didn't come to my brain immediately at all. So,
Yeah.
PIP3. PIP3 is fun.
Yeah. Oh yeah.
I had to remember
Pip.
PIP3. Yeah.
I remember Pip.
Did your exam look like this
Somewhat.
when you took this?
I think
No.
I
somewhat.
think we had a different.
I know he has to change it every time, otherwise we wouldn't even be allowed to do this.
Yeah.
So.
But the Darth Vader picture on it was the same.
Okay, good. At least there's a Darth Vader picture. But the rest of it,
Which question are you thinking?
a lot. There's some good ones.
there were
There's a lot of
some
good
good
ones,
ones. There were some decent things I did here, but not. It wasn't my greatest attempt in the world. I think I got like a median score or something. It
That's
wasn't
fine.
anything
We're
to,
gonna ask
wasn't anything to write home about, but
We'll talk about the question.
yeah.
Yeah, a whiteboard would definitely be good.
I was gonna bring it. I forgot. Sorry.
So there.
You guys get wavy hand motions instead.
Actually, you know what? Hang on.
Is that
I
a
think
whiteboard?
I
Oh,
have
you
a whiteboard.
can? Wait.
No, I think there's a whiteboard.
He's gonna go get a whiteboard real quick. So. Oh, I got 1 point for a sentence.
Nice.
Yay! I mean, that's pretty average nowadays. I mean, this would have been a good exam for me now.
Oh.
If I'd gotten this now,
Yeah.
better exam
I
probably.
like. You didn't draw anything.
I didn't draw anything. No, that was a bit worse. I got 4 points because I was somewhat close, but I didn't get the KD value.
Yeah. Yeah.
I did get somewhere close to the delta G value, but not the KD. I should have not considered initial and final. I should have gone with
Ooh,
some kind of a
another
Uh
visitor.
oh.
It's
We have
fine.
a poodle in the room.
Hello.
Hello.
That's good for the pod. People like dogs. That one doesn't have the ear infection.
That's good. Yep. Better.
There he goes.
In there. Sorry.
Is this camera still recording? Oh, it is.
Yeah. Would you look at
It's
that?
got a... Whoa.
Oh, sorry.
Technical difficulties. Okay, yeah, he does have a whiteboard. Hell yeah.
Told you.
We got it. Okay.
Have a whiteboard.
I guess we could illustrate the activation of protein kinase C.
Protein kinase C.
Okay.
PKC.
What does PKC do?
It. Oh, what does it do? Well, I mean, it phosphorylates downstream proteins
Oh,
to,
yeah.
activate things.
To activate things.
Give me this.
You know, you
What-
get that.
yes. What? Okay, so I was right about the PLC, whatever the PLC is. Then the PIP2 becomes the PIP. No, the PIP2. The PIP2 already became. The PIP2 came from the PIP3.
Yeah.
Yes.
So the PIP2
PI
phosphorylates PIP2 to generate PIP3. Okay, so you get
Yeah.
PIP3. What is PIP?
Phosphoinositol.
Right.
phosphate or something.
Exactly. This is crazy. He's already giving us like debris cells stuff
What?
already before we had done any DNA
Yeah.
or protein quantification. So this was a total shot in the dark. Seeing something like this when you've done nothing on it before, interesting, but I think it went okay.
Yeah, PLC is phospholipase C.
Right. He didn't give us any points that you could have gotten from this. He usually said 3 points or 2 points or something. That's the question. There's nothing here. It's just kind of the subjective, how well do you think you did on this?
Let's keep it to the biochemistry, I think.
Yeah.
This is the biochemistry. Look, it says literally right there, biochemistry, Chem 480, biochemistry. This is biochemistry.
Yes.
Oh, you mean
It's the grading.
grading
more.
stuff? Oh, well, grading is its own thing. You gotta be able to beat the system of grading. You got to pick, words that are good.
You got to know
That's
your
true.
content.
You gotta, it's
Pretty
usually
much.
how it is.
Yep.
Yeah, it's no longer just multiple choice.
All right, let's illustrate it. What are we illustrating?
But I don't want to illustrate it.
What does it say illustrate?
Illustrate the typical activation of protein kinase C. So the PIP2 goes to IPC. Look, I don't even know what these abbreviations really mean anymore. IP3. So let's go to PIP2. Guys, I truly studied for this. I hope you understand.
It's okay.
Okay. Good.
You want a hint?
Well, I'm just gonna write down exactly what I did.
I'll just give you a little hint, okay?
Okay, what's the hint?
You got to remember two things. Calcium
Mm-hmm.
and DAG.
DAG.
Yes.
Good old DAG. I remember DAG.
DAG.
Then what about PLC?
What about what?
It's a bad marker.
Oh, there's two more.
What do you
Sorry.
think PLC? What do you think PLC stands for?
Protein lipase C.
Close.
Phospholipase C.
There you go.
Phospholipase C.
Phospholipase C. What?
What is this?
Okay, try this one.
How to get?
If not, I have an orange one.
Dude, that is a rock. It's horrible.
Yeah, this one might be better.
Oh, yeah.
These aren't my markers, but I have.
Oh yeah.
Much
Dude.
better.
Look,
There you go.
there's cruddy marker. There's nothing marker, and then there's wonderfully flowed marker.
There you go.
Let's go with this. All right, so we got our little PLC right here. I got that right, so I'm gonna write that down.
Mhm.
Phospholipase C.
Yeah,
Do you remember what receptor it is?
this is a ligand. This needs a ligand.
What? Yeah. The ligand does bind to the receptor. Do you remember what type of receptor it was?
That does happen.
You have it kind of written here. What is that?
GSA, GS alpha.
What's that part of? It's good.
Think of it. It's in your brain.
It's
I
in
can
there.
feel it coming out.
We have the receptor tyrosine kinase receptor, then the G
Glucose.
no
Frick.
has G.
G. What is G?
Come on.
Gluc.
You're so close.
No,
It's not. It's just
There's
G.
nothing to do with gluc.
It's
It's
G.
G.
G. GSA. Gamma. No, I'm going farther away, aren't I?
It's the G protein receptor. Remember?
I don't remember. That's why I want to go over this. I've had a whole summer in between this and
G protein
geez.
receptor.
Yes. The GPCR. And so
Great.
the
This is why I don't do good on these exams. I don't just. I have a. I don't think practically at all.
interesting.
I don't think it's that.
So GPCR has extracellular receptor that binds to the ligand, and then on the intracellular side, it is made of GS alpha and G. I think it's G beta. Let me see. Oh, G gamma, and then the beta subunits, and so the gamma and beta subunits pop off, releasing the GS alpha, which
Well,
is.
that's great. You have all that on your phone right there.
I'm looking at it from a little bit off here. But it's the GS alpha is stimulatory, so
Okay.
that's what activates the PLC. You see
Yeah, you can take a look and then draw it out for
the
him.
gamma and beta subunits.
Okay, I'm gonna draw this instead because this looks way more familiar to me than whatever the heck I drew on this. So
You drew this, by the way.
Yeah, so far, if you can see that.
it's the forward reverse thing. So I drew this, but I also drew some other stuff like boo boo. PKC
Protein kinase C.
attached to DAG.
Yep.
Well, that's what he
Yes.
wrote right there. I didn't do the IP3.
Oh,
but
he filled that in for you.
I said PIP2 makes the IPC, IP3 and DAG.
It does.
And then the PIP2 forms the IPA PIP. I think I was writing the net equation or something right here.
Yeah.
The PIP2 then releases the IPA. IPA. What is an IPA?
Do you remember
It's a type of beer.
what IP3
Okay.
stands for?
But
Do you remember what IP3 stands for?
IP3 inositine.
It's actually not. No.
I
So your
Insulin phosphate.
no.
No.
Do you remember what it is, Norm?
No.
I don't remember.
It's the inorganic phosphate.
Inorganic phosphate 3. Oh. And then the inorganic
It's a weird
phosphate.
way of writing it, 'cause you usually just write PI. But
Yeah.
Sure that's right.
no, that is right.
That's how a biologist do it. Look, guys, I'm better at chemistry than biology. It's very true. Everyone knows that.
This isn't biology. This is biochemistry.
No, this is biology. This is
Mm.
what biology people write more of.
No.
This is why they were better at this stuff.
No, they're not.
Yeah.
Biology people draw glycolysis by putting circles on a page.
No. This
They
is
can't.
exactly how they like to draw things. This is schematic representation. This is not real chemicals or structures at all. This is just drawing. This is like drawing castles and
The difference is
wizards and
biologists
unicorns and
can't
stuff.
draw inorganic phosphate. They, just
They
write
probably
inorganic
can't.
phosphate.
No, exactly.
Yeah.
That's what we gotta.
They don't learn the structures very
Well, yeah. There's biology land, but before we get off to just
biology land.
before we start just
No offense if you
dunking on
love
it.
biology land.
Yeah.
just stay in biology land. But I just personally,
we
lose my
have
mind.
issues with biologists sometimes.
I would lose my mind stuck in biology land. I need more detail than that. You know,
We love you guys. We love biologists. You guys are great. We're just
some of you are great.
some of you are
I'm
great.
joking.
You're fine.
I'm
No,
joking.
you're fine, guys.
I'm
Don't
joking.
worry. You're good.
No, I'm not.
We all like
But let's say
you. Well, dang.
I'm just joking.
Dang, he started again.
I'm joking.
Okay. All right.
Just take a trip to Biochem land and see if you like it. I think you'll like it.
You will.
It's very involved.
Phosphotyrosine. Okay.
Are you done with this, the pathway?
We're done with the pathway back.
Oh, did you show him?
We're not done.
Oh yeah.
Oh, we're not
guys
done?
We're not done.
No, we are
You
done.
did it?
No.
Oh, you
You
missed
didn't.
it.
I mean, you didn't. So okay. So what else is produced from the PLC?
Calcium.
No. So
Cam.
PLC.
PLC.
Phospholipase C.
Phospholipase C.
Yeah, PIP2 and IP3 so far coming out.
Yeah.
What else comes from that? I thought we just talked about it, didn't we?
We just talked about it. Yeah.
Yeah, remember?
You have it on there, but you don't have it structure or like the.
I can't draw a structure of PIC. That stands for PIC.
PIC.
PLC.
Well, okay. So PIP2 releases
Ooh.
inorganic phosphate and what else?
Releases inorganic phosphate and
We just talked. I don't remember this. We just talked about it.
Let me cheat
What
here.
was it? Yeah.
PIP2? PIP2. No, PIP3.
Yes.
PIP3. Good old PIP3. I remember PIP3.
Colton saying no.
It's not PIP3. We'll say something.
It's DAG.
Oh, it's
that's where the
wait, but no,
DAG
DAG's
comes
up
from.
here.
Yeah, but like draw an arrow to it.
Oh, you want to draw? Oh, you
It's
mean
the-
like
it's the
that?
path.
Oh, no, that's why I was drawing this up here, is because we have a net equation for PIP2 making IP3 and then DAG, and then DAG goes up here, releases PKC.
It binds to PKC.
Right.
What else binds to PKC to activate? It's not just the DAG.
Oh, it did something else. PKC did something else.
Well, to activate it, it needs something else.
Calcium?
Yes.
Yes, the calcium comes up, activates
I
it too.
told you
Because that
about
opens
that.
up the ion channel. That's right. That's how this whole thing got back to it again. Okay.
Yes.
It's starting to come back to me finally, little by little.
They did it,
little.
guys. They came back to what I said.
They
5
came
minutes
back.
ago.
Okay, yeah.
Calcium and DAG. I did say that. I 100% said that. Not even Colton can deny that one. Mm-mm.
Okay.
Feel free to comment, person who's watching.
There's 2 people watching.
No, there's like one that's like some
No, there's 2.
bot or something.
No,
There's
one
2
did a
people.
thumbs up. There's 2 people watching.
Well, no, one. The other commenter gave the one up.
Yes, and there's 2 people on the stream right now. It says on the left. The 2 next to the person icon.
Who else is watching?
I don't know.
I don't
Somebody's
know.
watching.
If they want to just
Who cares?
they want to
They're
just
learning.
hang out, yeah, they're
Just
learning
hang out,
stuff.
yeah.
Yeah. So here's the whole pathway for people who are interested.
Sorry for the weird arrow up to DAG.
Take a
Also
take a
ignore the PIP3,
screenshot and
that's
look
not
at it
involved in
because
this
this is as good as you're gonna get.
Well, I mean, if they're gonna, you should erase the PIP3.
Yeah, because
No.
that's not actually,
No.
that's not a part of it.
That's not part of it. That's the
No
separate
PIP3.
pathway.
Yeah.
That
No
is-
PIP3. You guys are no fun. If you don't have PIP3, I don't know what you're doing.
Being
Oh,
a PIP 2,
Are
Or
you a
maybe
PIP2 or a
a
PIP3?
PIP 1, or maybe just a PI. I don't know. What? There's plenty of things you could be.
Yeah.
Why do you have to be a PIP 3?
You don't have to be anything.
That's true. That's true, Colton.
Are you a PIP 1 or a PIP 2?
Yeah. Good question.
So I should ask when you fail an exam, are you going to be a PIP 1 or a PIP 2?
I was just complaining, but it's fine. Teach their own. What other questions?
Oh, also there's a Darth. Let me.
Oh, show me Darth Vader.
Yeah, let me just. There you go. Darth Vader. That was on the exam. I'm not showing. I'm not showing any of that other stuff.
Yeah. Why so much biology slander? Biologist slander. There's no biologist slander. I just think there's a fundamental difference.
There's a big difference between us and
in biologists and biochemists and the way you learn this stuff, because we have to be more involved with the details and take the classes like physical chemistry that are terribly hard and risk our GPAs consistently, and you guys draw circles and say glucose. That's all. No offense.
That's our only gripe. I
Yeah,
mean,
it's
we need to find some way.
You know, perfectly fine to do that. I just find it annoying because it's not correct technically, but it's just me. Who cares? You should show them how to draw glucose actually really quick.
Oh yeah,
We can teach them all right now.
Absolutely. So guys, chair conformations, right? You have to do organic chemistry if you're a biologist.
Yeah,
maybe
the chairs.
Maybe. Do they?
No.
They
But
don't
we should
have
show
to.
them.
You guys don't have to. Okay, so you want to
makes
make
it
something
better.
like this. Really fun thing to do here. So if everyone can draw that, you're in the clear. It looks like a 1, 2, 3, 4, 5. It's a 6-sided thingy.
Well, I mean,
And
they
what
take
this is,
Pokin, so they should know how to draw that.
right?
That's
And what
fine.
this is this, and then this and this correspond to this and this. So where the two dots are, or where those things are on the hexagon, it's at a slightly angled, side view of that hexagon. So you can see like things pointing downwards. This part would be pointing down, and this part would be pointing up. Kind of like that. You can just kind of extrapolate. This is sorry guys, I know OChem is hard.
I mean,
It is hard.
eh.
Kind of like that. That's probably the best thing I can do
It's
for this.
different levels for different people.
No, that's true, but I mean,
Difficulties.
what do you mean by hard? I think Okim's more just a bunch of memorization.
Well, if we're talking
versus
about-
actually complex.
Well, you need to know air
need
pushing mechanisms
mechanisms
and
and
some of
But those are
required.
just memorization. That's what I mean.
That's
Well,
fair.
Memorization, but there's a way that you don't need to memorize all of them. You just need to memorize one way of doing something, and then you can kind of just remember. Oh, well, I know for a Claisen reaction, I need to do a carbonyl bray.
Wait, weren't
I
you
gotta-
arguing for it being hard though? Because now you're saying it's not that hard. I'm confused.
Well, no, it requires a bit more mathematically minded stuff, or
Okay, no, I see what you're
a systematic approach to things instead of just pure memorization.
Yeah,
Because in biology, you either know it or you don't, right? You either know how to taxonomize. Look, this is how bad with biology I am.
biology was pure memorization. Also, that's
It
why I'm
was.
I'd
confused.
say that one is more memorization.
Yeah, it's more.
So
Way more
Since we're
memorization.
talking about OChem.
Yeah.
Yeah.
Right? What does it do, chair confirmation?
That's true. Why does it?
Oh,
What does it do that?
that's a good question.
Yeah.
Why does it do chair?
Why does
Why-
it do
why
why it do chair
Why
conflict?
it-
It has to
Why
do with the steric hindrance, right, of the ring. The sterics make it kind of bend out. So you have, which one is it? Is it 3 and 1 or 3 and 5? One of the two is going to be up and 2 is down, or it's going to alternate and 2 is up and the other one's down.
why chair? Why chair there?
Yeah,
Stability.
it's stability. Yeah, but that has to do with steric hindrance.
It does.
That helps.
It can't just stay in the 6-membered ring formation because it's not stable enough.
You wanna,
So instead, it puckers.
Absolutely.
It's called a ring pucker. If you guys didn't know.
We're gonna do
Well,
the classic
is it not?
this one's not
equatorials.
ring pucker. That's a separate.
No, it's
step.
pucker.
Oh, that is you're right.
Pucker-
That's the, yeah,
so
yeah.
much
For like a
different.
pen-
Pucker.
pentane or
Excuse me,
something.
guys. Whoa.
No, you can draw the puckered hexane too. Ben showed us like the lazy boy chair though, where it's kind of straight
Oh.
instead. Yeah.
Oh, I know
That's
what you're
what I'm
talking
talking
about.
about, yes.
Oh, okay.
I'm talking about the puckered chair for that one. It
Well, yeah.
does it when you're breaking apart. Is it glycogen? One of them is puckered, I think. Or maybe it wasn't glycogen.
I talked about it with like 5-membered rings with the puckering. I don't remember.
You guys
I don't
see
remember.
that in
was
medical
fructose.
journals and stuff?
Fructose
Those two
had
are
a
puckered.
puckered state, yes.
They look
Versatility,
funny
yeah.
and
That
they're
makes sense.
cool,
Okay,
and
that's
I
what I'm
like to
thinking
draw
about.
them, especially on DNA. deoxyribonucleic acid. Here's a ribose. The ribose ring is kind of cool. It's like the fructose. It's the 5-numbered. This would be the ribose.
Did you draw a shadow?
This would be the glucose.
Wait, you never drew.
You draw ribose.
You didn't draw glucose. You just.
I didn't draw glucose.
Glucose.
No, I haven't.
thought
Look,
we were
we're going over chairs. We're going over very
Did you forget
basic.
how to
and
draw
small
glucose?
stuff. No, I have. I know how to draw glucose. I can draw glucose right now.
Yeah.
Heck yeah.
Because that was the whole point. We were going to show them how to draw glucose.
Right.
Yeah.
Exactly.
Mm-hmm.
And this one is OH. OHs are equatorial.
We should probably talk about equatorial
That's
versus
axial.
axial.
Yeah, we should do that. Equatorial versus axial. But I think you guys know equatorial versus axial. You're all smart enough here.
Yeah, you guys can do it.
I hope.
This is equatorial, right? Or is it like it's like that?
That's equatorial.
That's equatorial. So this right here is our good old friend glucose. Let me erase this one.
Oh, here. I brought this in here. So this is for
Oh,
a little eraser.
I thought it was your napkin
No, no.
or your tissue.
No, that's a different one.
I was just thinking, oh, might as well use this, even though you've got snot on it or something. Here we go.
Oh,
Good,
Jesus.
good. That's glucose.
Yeah.
Fun.
Oh, let's do a little trivia real quick since we have a second.
I'll call it GLC.
Here's a good question for you guys.
Yes.
If we were to phosphorylate glucose, where would it become phosphorylated?
Oh, yes. And which enzyme does it?
Yeah. Where would it become phosphorylated on here?
Where would it become phosphorus? So this is gluc, right?
There's a lot of OHs. Which one would be phosphorylated?
Which,
Feel free to type. Whoever's
type
in
in
chat.
the chat. But please, there's one person in the
Please.
chat.
Please type.
Please.
Yeah. Where
Please type
exactly
in the
would you
chat.
phosphorylate this molecule? It's okay if you're wrong. We'll just tell you.
Which O group could you phosphorylate? Which one could you put a
Those are called hydroxy groups.
hydroxy
just to be
group?
clear.
Oops.
Or alcohol.
Or alcohol. But
Where could you put
we'll
a
stick with
PO3?
the
Is it a 2
hydroxy?
minus? Is PO3 a 2 minus?
Yes.
Yes. Okay. Where could you put this guy? There.
Glucose is a blob. It doesn't
There.
matter. Jesus, this guy. Like, what?
Wasn't even watching.
I don't know. You got 3 options there. You got a bunch of options there. But which one do you think it's phosphorylated?
You should probably number the carbons
You
No,
should number
no.
the carbons. That'd
You're
be
right.
handy.
because they're just
See, there's. It's not a carbon.
nope.
I'm an idiot.
There.
1, 2, 3, 4, 5, 6.
Anybody know?
Someone needs to know.
I know.
Well, he knows.
Well, there's 2
Well,
places.
how about we
that
give
is-
it? How about we give him the enzyme? The enzyme that does it.
Oh, yeah, go ahead.
Hexokinase.
Yep, hexokinase.
Hexokinase.
Or its more common name. You guys know the common name?
glucose 6 phosphorylase.
Yes.
Oh, good. What'd I do? What'd I do?
Glucose
GLC
6 phosphorylase.
phosphorylase.
GLC 6P.
Yeah, you fit that
Oh
in.
my gosh, I'm ruining it. Phosphorylase. Here we go.
You could just show
This I literally gave it away. So, plug 6 phosphorylase.
Yeah, you look at the name of the enzyme and you can tell where it goes, right?
Yeah.
It's on the 6 carbon.
6 carbon. So you erase the H on the 6 carbon and you now have OPO32 minus.
It doesn't go to any of these. I'm making a Dr. Seuss
You guys remember
illustration here.
why it gets phosphorylated and when it gets phosphorylated?
Wait, what'd you say?
Why and when the glucose gets phosphorylated?
Why it gets phosphorylated and when?
Yeah.
It's going down glycolysis to become acetyl-CoA.
Okay. Well, does it? Is that all?
What do you mean is that all?
Is that all the only reason?
No, you start phosphorylating it when insulin's high because you need energy. Then you start going down glycolysis. What are you asking
Now
specifically?
you got
Well, it's not the
glucose
only
6P.
reason. So,
There you
the
go. That's the
glucose
second
going
step
into
of
the cell.
glycolysis.
Yeah.
is in
No, it's
equilibrium
the first step.
with it leaving. Right.
Yes, that's why that's a regulated step.
Exactly. So when the glucose enters the cell, you phosphorylate it
Mhm.
so it can't go back out.
Oh,
Which
yeah, you
pushes-
trap it in and force it to go down glycolysis. Yes.
It
That's-
pushes the equilibrium towards going into the cell
Yep.
since it can't leave. And
Guys, we've been
Yeah.
streaming for almost an hour.
Yeah, that's the goal.
That's really great stuff.
wow.
We can go over time just a little bit, but we got Mongolian to eat pretty soon. So
Yep.
we got
We do got
we have lunch awaiting ahead of us.
That was pretty good, though. We owned a lot of stuff.
I don't remember if I locked my car or not. It's probably okay.
Oh, up here it's fine.
It's
Yeah, no,
Valley's very safe, guys.
for the most part. Downtown, not so much. Anyway, it's not the point.
That's fine.
Well, sadly, nobody actually knew the answer to the question, which is
That's
disappointing.
crazy, yeah.
Because you asked a question about biologist slander, then continued to slander yourself by not knowing the simple answer. But that's fine, you know. You learned something today.
Mm-hmm. Well, we're not gonna.
And we love biologists.
We like you guys. It's okay.
We love biologists to death. We're just more chemistry oriented, especially Colton.
Well, I like both. That's why I did biochemistry.
Did you like genetics?
Yeah,
Genetics was interesting. You didn't like genetics?
I loved it.
I didn't like the professor, but I mean,
we don't need to slander professors.
We're not gonna slander. I'm not gonna say who I took it with, but you know.
But if they watch it, they know who you are.
Oh crap!
Yeah, so you just kind of set yourself up for that one.
Oops.
Yeah, we love all the professors.
We like.
But no, genetics was interesting.
Yeah,
the class was kind of hard because I did accelerated, but it still wasn't bad. It was like you actually enjoyed it. Learned a lot, and now we're doing the same thing, genetics, but not really, just
the biochemistry.
Yeah, versions essentially. And I will say, genetics made this class a lot easier because most of the stuff I already know, transcription and RNA polymerase and all that. We learned about it, so you got the background.
Oh yeah,
locked into your brain. What are you doing?
You trying
scribbling.
Oh, is that glucose?
Yes.
Oh.
Guys, this is glucose, by the way. You can make it look like that.
You're not a biochemist, yes?
That's fine.
Kind of.
Just a biotech.
Is that the only whiteboard?
Yes.
No. Do you want to draw?
7.
Well, I was gonna see if we should probably draw the other pathways.
Cool.
Just for the other.
Knock yourself out.
People.
Yeah, go ahead. You can draw some more pathways. I'll bring my whiteboard for the third episode.
Plates
it's way bigger.
I have plates to bring out of the oven at 5:30ish.
Oh, geez.
Yep. And the incubator. We're still doing research over there. So.
Yeah, you gotta keep doing research.
You have stuff to do today?
No.
Okay, good.
Me and Kyle ran ours into Thursday.
Yeah.
I could go in and
I took the stuff out for you guys.
Make some more crystals, but I think I'll just wait till tomorrow because I need Jackie for
my Yeah, Dr. Ben
HF.
and I just took it out.
He's there right now?
No. We took it out on Friday or whenever you told me to take it out. So it was pretty fun. We went over, we took it out. It looked pretty good.
We got another message. Hey, that's glucose when you drew it. Wow. I'm glad you enjoyed his drawing of glucose.
Yay.
Yeah. Jesus. Anyway, it's fine. Colton here is drawing you guys some more pathways.
Here he goes.
To increase your knowledge. George is just taking a nap, enjoying the biochemistry talk. He might have been a biochemist in his past life. I don't know.
Is he just pure chemistry now?
Who?
Never mind. Let's not name names.
I said George. I was talking about the orange cat laying.
Oh, the George.
Who did you think I meant?
Never mind. Never mind.
Yeah.
Let's not name names.
No. Antimony L element. All right, bud. That's cool.
Antimony.
I would ask you what your favorite element is, but you're a biologist. You don't even know them. So that'd be a waste of our time. Anyway, look at this sick pathway.
I'm not done with it.
Well, he's
No, he's still working hard on it. Sorry.
He's got this in the...
Let's let him work pretty hard here.
Oh, yeah. He's got this in the bag. He has this right on the tip of his tongue. He's got this GSA.
Which one is this one?
This is the adenylate cyclase.
Oh, okay.
Oh,
Remember that?
I remember that one, yeah. I remember adenylate cyclase. I remember most of the stuff.
Yeah.
Yep. Pretty sure. You might just have to tell me some names. Do.
Oh, yeah.
Do. How's the MCAT subreddit going? I look at that from time to time. Sometimes.
I don't. It gets annoying.
Well, it's mostly people either flaunting their scores or saying they had absolute rotten luck.
Or making up their scores.
Maybe. Well, they have. Oh, yeah. Here's a good one. This person got a 522. That's crazy.
Oh.
Here, look at this.
That's a good score.
Yeah. It's not like mine. I got a 495 on the MCAT. It's very poor. But I'll work at it to get a better one.
I got a 522 working 50 hours a week.
Wow. Goodness.
It's a lot of work.
That is a lot of work. This is somebody who knows what they're doing. I clearly didn't know what I was doing.
No, you didn't do bad.
Well, I mean, yeah, for not studying, I mean, a 495 is, probably what I deserve, but that's okay. Goodness sakes, 520. That's another 522 word. I want to see somebody who got closer to my score. You can say what MCAT score you got, right?
Yeah.
Oh, of course you can. Yeah, I would think. I know my uncle told me what his was, and it was on a totally different scaling. It was
back then.
Yeah, it was out of like 70 points or something really low.
Yep.
It wasn't.
That's how the dad is. It's really low scoring, too.
Yeah, that also.
Okay.
You ready?
Yeah.
Oh, look, Michael, you got the pathway.
It is.
Yay!
Yeah. Hold up.
Cool.
There you go.
That's a brand new pathway for everybody. That's
adenylate cyclase.
That uses GTP. That's guanosine triphosphate, not adenosine triphosphate. Don't get those mixed up. But very slight difference between the two. One is ketone, the other is an amide.
What's the point of this pathway?
Point of this pathway is to make.
You should tell them. What's the point? What are you making?
To activate PKA.
Yep,
Activates PKA.
You also get camp out of it.
And yep, a CAMP, which activates the PKA.
PKA, not to be confused with PKA of amino acids or something. Where the pH at which
Protein
comes off.
kinase A.
Yeah, it's protein kinase.
Yeah.
Two different PKAs. That's something he always wanted to hammer home about.
You got to know it.
Sure do.
All right. We're at our hour. That was about perfectly fun. Yeah.
How I feel when I think I fully understand a car's passage and then get to the questions and miss every single one.
All right, guys. Well, thank you for watching the second episode of Past the Porin. It was fun. Hopefully you guys got some knowledge out of that and actually learned how to draw glucose. That's great. Anyway, it was fun. We have lunch to eat, so we will see you guys next week for the next episode.
It was fun. Thank you, guys. This is Ben. Your name?
Oh, Nori.
Michael.
Colton.
I thought it was a catch line. I was supposed to say something.
No.
I'm so confused.
No. Yeah, just all right. We'll see you guys next week. Hopefully, all three of us will be here again.
We'll have some new content, and I'll put out another poll this time.
Yeah.
To see if you can pick an article that you want. Best stream ever. Oh, thanks.
Oh, thank you.
Mason Cundy.
Oh,
Thank you, Mason.
That's phenomenal.
All
I
right.
appreciate you watching and engaging with us. All right. Anyway, we'll see you guys later.
See you guys later. Bye-bye. Bye-bye.